right, good morning everyone, and thank you to the organizers for the opportunity to present some of our work here today. Fingers crossed, had a few issues with the AV setting up, but hopefully everything runs okay. All right, I have to make this disclosure. My university requires this. It's just to say that some of the technology I'll be showing you here today has been commercialized. We have a startup company, Arikari, who's also here at the meeting, so I'm wearing two hats, but right now I'm wearing my academic hat. So all good there. Just put this up. I'm using this a lot recently. It's kind of a validation for what we're all doing. We've been working in this field for probably 10, 15 years now in microphysiological systems, and hoping that the day would come when the FDA got all excited about it. And now they have. It's in there. Organ chips, microphysiological systems can be used for submitting INDs and the like with the FDA. So I think our time has come, and that's pretty exciting. So what I'm going to talk to you about today are the base platform that we developed, which some of you may have seen before, which we call a vascularized microorgan, or a VMO. And then I'm going to talk about two different vascular malformations. One is in the title of my talk, which is HHT, or hereditary hemorrhagic telangiectasia. And the other is the new data we have on port wine birthmark. So starting off with our base platform, I always put this slide in, you guys know this, but it's always good to emphasize just the huge difference between what a real tissue looks like with multiple cell types in a three-dimensional organ and where we used to be with monolayers. We and others have developed vascular systems. I work in vascular biology, and a couple of examples there. And the reason we do it, a couple of data slides I'm going to put in here. The assay doesn't matter, but basically what you're looking at here are dose responses, and this red dotted line is the IC90. So you can see these drugs inhibit this process very nicely in this 3D platform. The exact same cells in 2D, and these drugs at the same concentrations have absolutely no effect. So to see the effect of these drugs, we need to use a 3D platform. And here again, this has just happened to be tumor growth in one of our platforms. Happens very nicely. Very sensitive to this drug at all concentrations we tested. If you put those exact same tumor cells in 2D culture, they are 100% resistant to the drug. So a couple of examples of why it's so important to use these 3D systems. And so we need 3D. We are strong believers in human biology. That's what we're all interested in. So as much as possible, we want to use human tissues. I'm a vascular biologist, so everything begins and ends with blood vessels. You're all in this meeting, so hopefully you agree. And if you're going to have vessels in your tissue, you might as well have them perfused as well. And so what we've created is this vascularized microorgan, which we like to think sits in this sweet spot between the simple but not very physiologic monolayer cultures we've all been used to, and the very definition of physiologic but hard to use, which is the human. The only time we can use those for studies is in clinical trials, expensive, difficult to do, and not without risk. You'll hear a lot here about spheroids. These have been very useful. There's a lot you can do with them. I'm not going to show you data today. We have data to show that in many cases they're not as good as the three-dimensional systems that we're going to talk about. Other times they are useful. So as always, your model should be fit for purpose. It should be as complex as you need to answer the question you have, but no more complex than that. And of course, there's mice here. We've done a huge amount of work in mice over the years in the field. Incredibly helpful model. But again, to everyone outside of science, these two things are very different. Scientists tend to see mice and humans as largely interchangeable. If you ask a person on the street, is there a difference between a mouse and a human, they're just going to laugh at you. Well, of course there is. There are huge differences, and we shouldn't ignore those. So this is your vascular system you're all familiar with, high pressure arterial side, a low pressure venular side, connected by a capillary network. We schematize it this way with a microfluidic artery and a microfluidic vein. And we have this diamond-shaped tissue chamber that connects the two. And within this chamber, we create a capillary network. And that's what you're seeing here in red are human endothelial cells. There's the artery, the vein, the capillary network. And around this, we can grow any tissue we want. So we put a matrix in here with the endothelial cells. And then in the background, which are normally not labeled but are always there, are stromal cells, which are important for developing the vasculature, and then any tissue-specific cell we want. And that could be heart, liver, pancreas, brain, tumor, etc. So it's a very flexible platform. The vessels form spontaneously over time. This is a biology-directed process. There's no pre-patterning. This is our original version. I just put it up because it just emphasizes how flow through this system is driven by hydrostatic heads. You're all familiar with that, I'm sure. So flow goes from V1 to V2 along the artery, V3 to V4 along the veins, so that's there to there. And because this side is higher than that, the net flow is always in the left, top left in the artery, through the vessels and out the bottom right. And by changing the hydrostatic heads, we can control the flow rate through these vessels. This has all now been collapsed down into 96-well plate format, and we can increase the depth of these wells again to control the flow rates. And you can see how it all lines up very nicely in there. So it's now a relatively higher throughput platform than this original version. So why do we go to all this effort to put in blood vessels? So all organs are vascularized, as we know. Vessels actually help to pattern tissues. There are several organs now where it's shown there's an actual crosstalk between the endothelial cells and the local tissues to help pattern that tissue. Most cells are within 100 microns of a blood vessel, and all nutrients and most drugs are delivered through vessels. Immune cells, if you're interested in those, also delivered through the vasculature. Mice are not people. And so to best model human disease, we need vascularized, perfused human tissues. So again, this is what it looks like. We have endothelial cells of various sources, stromal cells. We begin with some fibrin in the matrix. That rapidly gets remodeled and turned. The matrix becomes mostly collagens and laminins. Here you do see the stromal cells in red in the background. As I say, mostly they're not labeled. This is just what the vessels look like, the artery and some capillaries coming off there. Here we're running through a red dye, and you can see that flows through the vessels. These are beads to show there's flow. We can flow blood through, and when we do that, it looks very much like blood flow in vivo. There are areas of high flow, areas of low flow, and even areas here and over here somewhere where the red blood cells have to squeeze through very small vessels. So in all ways we can see these look like vessels in vivo. They are wrapped by pericytes. You can see here they are polarized, collagen 4 base membrane, ICAM on the luminal surface, and again here you can see these endothelial cells are nicely polarized. We have looked at permeability. So 70 kilodalton fits each dextran, the size of albumin, leaks slowly. Something size of an antibody leaks more slowly. And if you challenge these vessels with something like thrombin or LPS, they become leaky. So again, they are like real vessels, we believe. So what we've done with these is to look at a disease called HHT, or hereditary hemorrhagic telangiectasia, which is obviously a bit of a mouthful, so we'll just stick with HHT. It is a vascular disorder. At the small scale, you get these small lesions on the skin, the lips, high traffic areas, as we say. These are called telangiectasias. But you can also get these larger AVMs in the brain, the liver, the lung, and they can cause problems if they burst. There are also issues with redirecting blood. So we know the mutations that drive HHT. They're in endoglin and ALK1 or ACVRL1. These are receptors involved in TGF-beta BMP family signaling, especially BMP9 and BMP10. And we know that these pathways have a role in vascular development and remodeling and integrity, but what we don't yet understand is why mutations in these genes cause telangiectasias and AVMs. So here we have an artery and a vein, and under certain conditions, inflammation and VEGF, in the absence of ALK1 or endoglin, two things happen. You get vascular overgrowth of these small vessels, and you get formation of these larger AVMs, arteriovenous malformations. So that's what we're going to try and model. To do that, we're using an inducible SH knockdown. So this is basically driven by IPTG, a small sugar, just removes the lac repressor of this promoter, allowing shRNA expression. By changing the concentration of IPTG, we can dial in the degree of loss of this gene. Without going into details, that's important. And this is just showing that IPTG itself has no effect on the vasculature. And so when we do indeed knock down ACVRL1, what we see is an overgrowth of vasculature, a formation of a telangiectasia that's quantified here. In this platform, it's relatively modest. As we'll see, we've got a modified platform in the next few slides where we get a more dramatic effect, but we can quantify this, we get nice significance of this generalized overgrowth of the vasculature in the absence of this gene. So while this was going on, I was working with the charity that helps patients with this disease. They're called Cure HHT. And based on some earlier studies, we suspected that anti-angiogenics might be effective in these patients. And so we set up a clinical trial to test pazopanib. That's a VEGFR2 inhibitor that's been used in oncology at a relatively high dose to block angiogenesis in tumors. It's widely used now. So we started to test this in patients at a much lower dose, and it turned out to be really quite effective in preventing bleeding in these patients. And for most patients, that's the major problem they have. They have a lot of nosebleeds, and you've all had a nosebleed in your life, I'm sure. It lasted a few minutes, and it looked like a lot of blood. It messed up a whole bunch of tissues. These patients, when they have a bleed, it lasts half an hour, and they can lose half a pint of blood. Very messy, very socially challenging, because it can happen at any time. And that's what patients want fixed. The AVMs cause longer-term problems, but on a daily basis, it's this bleeding that they'd like fixed. And pazopanib really helped with that. There was a follow-up study as well. So that was really exciting. And we had done some work in one of our other platforms earlier on, which we think shows how this mechanism is operating, how pazopanib might be operating. And so what we found was that BMP9, this is the ligand for ALK1 endoglin, we found that it induced this transcription factor called HESR1. And in a previous study, we had shown that this transcription factor downregulates VEGFR2 expression. So one might expect in HHT, where you lose the receptor for BMP9, you would think that VEGFR2 expression might go up, and indeed it does. If you knock out ALK1, VEGFR2 goes up. And then if you look at sprouting, vascular sprouting, under wild-type conditions, there's a very nice dose response of VEGFR2 to VEGF, and this isn't really reported very much. But at low doses, you get a nice peak in sprouting. Turns out higher doses, you get wider vessels. But what we found was when you knock out ALK1 and get more VEGFR2, you get an increased sensitivity to VEGF. This is just a schematic of what we think is going on. And so what we believe is that the loss of ALK1 leads to an increase in VEGFR2, and therefore an increase in sensitivity to the inhibitor of VEGFR2. And so this is the different platform that I talked about. It's sort of bigger and wider and better. And so here, when we knock out ALK1 with IPTG, again we get this vascular overgrowth phenotype, and then if we add pazopanib, we can inhibit that. And here we've got what we call a lesion score, so when we knock out ALK1, you can see there's a nice increase in lesions and telangiectasia formation, and then at higher doses of pazopanib, you can see we get a nice decrease in vascularity. So again, we can recreate what's happening in the patients on this chip. What we didn't see here are AVMs, the larger formations. And so what we think might be going on, when you look at the retina, and this is a nice model for vasculature, there are high shear areas and low shear areas. At the edge is low, here it's higher shear. When you knock out ALK1 in the mouse and look in the retina, what you find is at the edge in low shear conditions, you get overgrowth, like we see, but in these high shear areas, you get AVM formation. And so what we thought was we need more shear, and so we redesigned the platform into this bigger version, gave it higher flow rates by increasing the hydrostatic head, and this is COMSOL modeling of real vascular networks. And now in this light blue, that's the level of shear that we believe is necessary for the formation of AVMs. And so we think this platform should generate AVMs. So the first thing that we noted when we used this modified platform is we looked like it was getting arterialization of the arterial, which is, this is the arterial, this is the venual, and when we stained for alpha SMA, what we found was that our stromal cells were differentiating from a fibroblast pericyte-like phenotype into a more smooth muscle phenotype expressing alpha SMA, and those cells migrated to the artery and wrapped around the artery, whereas they did not go to the vein. And then when we extracted either the artery or the vein from our platform and did qPCR, you can see a strong induction of ephrin B2, which is an arterial marker in the artery, but not in the vein. So it looks like we are arterializing, and so again, should be a good chance we can generate an AVM, and that is exactly what we saw. So when an AVM forms, all the collateral vasculature collapses because of a loss of flow. And so while we have what looks like a nice vascular network here, when we look at flow, it's only going through this single vessel here. So this is the wild type. You've got a nice perfused network. Here, when we knock out ALK1, we now have this single AVM, arteriovenous malformation, and this is just quantifying the number of those. Again, here are the real vessels. This is the COMSOL simulation, and here you can see we're getting those shear levels that we believe are necessary to drive this phenotype. And then once more, again, you can see this nice AVM forming. So we've now done single-cell-seq on this. So this is all the tissue. So the endothelial cells are here. The stromal cells are here. These are the smooth muscle cells. When we mark these relative to whether they come from the control or the knockout, we find this population that's almost exclusively from the knockout. When we just group and cluster on the endothelial cells, we get 4 distinct clusters of endothelial cells, and it turns out that cluster 2 is almost exclusively from the knockout cells. So this is very new data. We're now doing cell chat and all the rest of it to look at cell-cell interactions and try and identify some key genes that may be downstream of the loss of ALK1 that may relate to how these AVMs form. So that's currently ongoing work. And in the last few minutes, I just want to talk about this other vascular malformation that we're working on. This is called port wine birthmark, and it's driven by an activating GNAQ mutation. It's not pretty, so if you don't like looking at stuff, close your eyes now. So this is a simple lesion, but you can see in extreme cases, it's really grossly disfiguring for these patients. Okay, you can look again. And so, as I said, it's an activating mutation in GNAQ that then drives multiple downstream pathways. It's not been worked out yet which of these are critical, but all we know is that several of these do get activated. And so what we did, we took an iPSC line that we had previously shown could be used to generate vasculature, and I'll get onto that in a moment. And then we used CRISPR to put in this point mutation into GNAQ. Then we crossed our fingers that we could actually generate vasculature from those, and we can. And you don't see it so well here, you'll see it in a moment, but all of these vessels are distinctly wider than all of these vessels. And what we're looking at here is perfusion, so we're putting in fitsidextran. It is perfusing here, you're just not seeing it very well. Because these vessels are wider, it's going through faster and greater levels, so we're getting nice perfusion, so they're great vessels, they're just fatter than normal. And if you leave these for a while, these are the wild type of regular capillary bed of narrow vessels, but with GNAQ mutation, you can see we get these hugely dilated vessels. That's exactly what you see in these patients. That's why the skin is red. It's because of all the vasculature in there and because of these large dilated vessels. And some of those, as you saw if you had your eyes open earlier, grow into these really large nodules under the skin. So we can quantify that, and you can see the GNAQ mutant in red. We get a lot of these much larger dilated vessels. So where are we going with all of this? What we want to do, because these are rare diseases, is repurpose drugs. As we know, pharma, for obvious reasons, are not particularly interested, most of them, in developing drugs for rare diseases because, by definition, there is no market. So what we in the rare disease community like to do is try and find a drug that's already been approved for one use and repurpose it for the rare disease. That's what we did with pazopanib. It works for some patients really well, but not for all. And so we would like to do screens on these platforms, and we're just starting those, to find other drugs that we can repurpose to give to patients. For HHT, we'll have to do animal studies with those, and there is an HHT model. Port wine birthmark, you can use topical application because it's a surface lesion. So there's a much lower barrier to entry with FDA for that. So we're going to be looking at various drugs, see if we can get one that'll work on that. Then obviously try and do trials. What we and many people are interested in are patient-specific models. And the reason for that is that we have many cases in the HHT community where patients with the exact same phenotype have dramatically different disease. One could be a 5-year-old who has terrible bleeding and dies of a brain bleed. And then there could be a 67-year-old with the exact same mutation. Yeah, I had a couple of nosebleeds once in a while, but that's it. So we now know that the genetic backgrounds of the patients is very important in determining what level of disease they get. So to understand that, we need to have those background genetics. So we're making patient-specific iPSCs. And that's all well and good, except not all iPSC lines in the past make good endothelial cells. And by that, I mean endothelial cells that will make blood vessels. You can very easily turn iPSCs into endothelial cells that grow as nice monolayers and express CD31. But you ask those to make blood vessels, and mostly what you get is this. They'll kind of make them, but they'll be massively leaky. They won't anastomose properly. What we've now done, thankfully, is found some protocols that we can turn pretty much every iPSC that we've looked at into really good endothelial cells. Part of it, which is published by others, is a pulse of ETV2. That helps, but not always. But it turns out just subtly tweaking the differentiation protocol actually makes a big difference. And so we can now and are collecting cells from patients, making iPSCs from those and then differentiating them into endothelial cells so we can do patient-specific models for HHT and potentially um, port wine birthmark. So to summarize, tissue microenvironments are complex. We need 3D models. As you know, mice are not people. And it's finished. Hang on, my timer's gone off. Anyone know how to stop a timer? Okay, there we go. So we need vascularized tissues, we believe, in most cases, so that you can understand the complexity of a tissue. Obviously, if you're working on vascular malformations, you're going to be working on blood vessels. And the final and most important slide, as always, is the wonderful people in our labs that do the work. All of these people are working on super cool models. Jennifer Fang here developed the HHT model. She's now started her own lab at Tulane. Will van Treit up here did the port wine birthmark model. Others helped develop the basic platform. Always big shout out to my collaborators, particularly Steve George and Abe Lee, who helped to develop this platform and continue to work with those. NIH, as always, for their foresight in funding us on these platforms for so many years. And particularly to Cure HHT, the patient charity, who I work with a lot and helped me get the IPSCs. And with that, I'd be happy to take questions. Thank you. I'm not sure we have microphones. If you have a question, you have to shout. Or not. All right, cool. All right.
Okay, thank you very much for your nice talk. You can stay here for the Q&A. So, if you want to stay here for the Q&A session. There is space for some questions, so the floor is open for question and answer.
Thank you for your talk. I was wondering, you mentioned about the fibrin being substituted with collagen by the time. So have you actually quantified that? How long does it take to get rid of the fibrin, or how long will the process go?
Yes, I have, and I put that slide in and then took it out. So let me just show you that. Yeah, here it is. I can't use a PC. How do you right click on a PC? Is it Sturge? How do I show that slide? No, it's hidden. Oh, there it is. Okay, yeah, that works. Thank you. So yeah, we did second harmonic generation, which shows collagen fibrils, and so you can see here over time how we get this massive increase in collagen. And there's a lot of plasmin expression in the early stages, so that's going to degrade the fibrinogen, so the fibrin. So we do get remodeling. We've also published a couple of papers, Newman et al., 2 papers around 2012, 2013, where we've done a lot of protein mass spec, looking at what proteins are actually made. So we've got a whole list of matrix proteins, and it turns out the key ones are matricellular proteins, things like beta IgH3, SPARC, IGF-BP7. Those are made by the stromal cells and are absolutely essential for vessel formation. And if you knock those out, you lose lumen formation. If you add them back, you get the lumens form again. So the matrix is really actually quite critical. And then you can see just laying down of a collagen 4-rich basement membrane over time as well. Thank you.
Any other questions?
Hi, thank you for your talk. I was curious about your single-cell RNA-seq of your HHT work. So you were saying something about a change in the EC population. Did you also look at the mural cell population? Did you see any changes there?
Good question. There are differences. Yeah, we haven't delved deeply into that yet, but one of the guys on that picture, Chris Hatch, he's doing all the stromal work. So every time we do a new platform, he gets the data to look at the stromal side of it. And as I mentioned earlier, in all tissues we've looked at, there is crosstalk between the vessels and every other cell in there. So most of the dramatic changes that we see, not surprisingly, are in the endothelial cells. But there are definitely changes in the stroma when we knock down ALK1 in the endothelial cells, yeah. Great question. It's something we're really interested in.
Thank you so much. That's all the time we have for questions. If you have more questions, you are welcome to follow after this session.